Hello, everyone, and welcome to today's webinar, Early ctDNA Molecular Response Enhanced through Integrated Analysis of Free Cell DNA and Matched White Blood Cells. I am Kaylee Bach of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by LabCorp. To learn more about our sponsor, please visit oncology.labcorp.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. With that, I'd like to now welcome our speakers, Dr. Mark Sausen, Executive Director, Head of Technology Innovation, PGDX LabCorp, and Elsa Anagnostou, MD, PhD, Associate Professor, Thoracic Oncology, Biorespiratory Director, Thoracic Oncology Precision Medicine Center of Excellence Co-Leader, Molecular Tumor Board Director, Sidney Kimmel Comprehensive Cancer Center, Johns Hopkins University School of Medicine. Mark, Elsa, welcome. You may now begin your presentation.
Yeah, thank you. So great to be here. I'm Mark Sausen, Lead Technology Innovation within PGDX and LabCorp. Excited to be joined by Elsa today as well. And we'll be focusing on how analyses of early ctDNA molecular response can be enhanced through paired analyses of cell-free DNA and matched white blood cells. So I think, one of the reasons, certainly one of the reasons I'm here today, and I think many of you that have joined is, a lot of the excitement around how liquid biopsies can be utilized to really transform how cancer patients are managed. And that's really all the way, from, how ctDNA could be utilized to detect cancers, characterize tumors, in order to better understand, the specific alterations that the patients may be the most responsive for, as well as for monitoring cancers, surveillance applications, and really a number of data sets that have now come out, both retrospectively and prospectively, for how ctDNA could potentially reflect tumor burden dynamics earlier than can be achieved through conventional diagnostic modalities. And this is particularly exciting to for settings where we're interested in capturing tumor burden dynamics during therapeutic intervention, which would potentially allow for patients with primary resistance to be rapidly identified, potentially redirected towards alternative therapies. But as over the past 5-10 years, as we've started to increase the size of these panels, largely due to decreases in sequencing costs and improvement in error correction based technologies, we've learned some of the biological challenges associated with the accurate classification of circulating tumor DNA specific alterations within the cell-free DNA. And that has the potential to confound these analyses for non-invasive tumor profiling, molecular response assessment, and clonal evolution, particularly as we start to expand our interest for, instead of individual biomarkers towards, all biomarkers or all alterations that may be detected across a large gene panel. So I'll be focusing initially on some of the analytical and platform-based approaches for how we've approached the accurate classification of variant origin to inform these different types of analyses for different cell-free DNA applications, and then turn it over to Elsa to highlight some of the results from her recent studies looking at longitudinal analyses of ctDNA for monitoring therapeutic response during immune checkpoint blockade, and ultimately some compelling data for looking at associations associated with clinical outcomes for these patients. So taking a big step back, as we think about how cancer care has improved over the past century, we've really gone from thinking about cancer as a single disease with a one-size-fits-all approach, where patients are largely being managed. We're largely being managed through surgical treatment, radiotherapy, and chemotherapy in an unstratified fashion. Towards the past 50 years or so, with the emergence of cancer-specific therapies, where patients were being stratified based on disease, and then ultimately with the emergence of targeted therapies, where patients were then stratified based on individual biomarkers within a disease, and then finally, as we think about the stratification of patients based on their broader biomarker profile, and how we may be able to leverage further improved drug development opportunities for these patients for individualized applications. And so I think this is a monumental achievement for really a lot of you that on the drug development side that have led to the development and ultimately improved efficacy of these different therapeutics over existing standard of care. And now it's really our job to really understand how we can further enable broader-based testing to ensure that the right patients are receiving the right therapies, given only 15 to 20% of patients right now are receiving access to what we would consider the minimal requirement to ensure the appropriate standard of care is identified for a patient, or if a particular clinical trial may be most appropriate. And so this is really an area where we're excited about for liquid biopsy-based applications. I'd like to highlight this seminal study led by Erica Carpenter's group, several years ago now, but really asked a simple question for does adding plasma-based sequencing to tissue next-generation sequencing improve mutation detection for patients with non-small cell lung cancer? And so this was a single-center cohort prospective study looking at 323 patients. 229 had concurrent plasma and tissue next-generation sequencing, and then tumor tissue-based assessments were able to identify targetable mutations for 47 patients, which was about 21% of the population. But the addition of plasma-based sequencing increased that from 21% to about 36%. And then of the 42 patients who ultimately went on to receive plasma-based next-generation sequencing indicated therapies, 86% of those patients were able to achieve a complete or partial response or stable disease. It's highlighted in the waterfall plot on the right, and really highlighted and concluded that adding plasma-based next-generation sequencing to the routine management of metastatic non-small cell lung cancer, has the ability to increase targetable mutation detection and really improve the delivery of targeted therapies. So very compelling and early data to highlight how plasma-based approaches can be used to complement tumor tissue-based assessments and really try and shrink this gap for where we are today for where we would like to be in order to ensure broader access to these different types of diagnostic approaches. And so within Labcorp Oncology, we really share the broader vision of trying to bring the promise of precision medicine within the reach to empower better outcomes for all. And that's achieved through these 4 different key pillars. The first being leading scientific innovation. That's thinking about how we can develop these liquid biopsy technologies for specific applications. But it's really also how can we partner with our drug development colleagues to through their proof of concept or registrational studies to ultimately empower better outcomes, demonstrate improvements over the existing standard of care. And then, once these, are out there for patients, ensuring that, patients have access to these different diagnostic, approaches to ensure that, we're able to, identify the right patients for therapies that they're most likely to benefit from, and then really trying to, do that in an impactful and positive way, in the context of their broader care that they're receiving. And so we've developed a broad, comprehensive portfolio of products to assess, all the way from risk assessment through cancer screening, diagnostic and prognostic testing, predictive testing, and then, ultimately standard of care treatment and monitoring or clinical trials. And so for the purpose of this discussion today, we'll be mostly focusing on the right side of this, which is once we've identified the appropriate standard of care treatment or the appropriate, clinical trial, how do we ensure that, as early as possible, we can identify patients that are responding or aren't responding, and then, what are ways in which those patients could potentially be better managed, and, identify more appropriate, therapeutics that patient may benefit from. And so from a testing perspective, we've developed a novel, dual-pronged approach to how we can enable groups to have access to these different types of diagnostic strategies. The first on the left side is highlighted through centralized testing. Certainly for services through our biopharma partners, but also for routine clinical diagnostic testing. Where samples are received, and then we're able to generate reports and provide those back to the appropriate groups for investigational use or for clinical use, to ensure we're getting the right information back for patients and their care teams. And then on the right is the decentralized strategy, or the kitted approach, where for these same types of technologies and applications, we're able to provide kits that can then be internalized through different clinical or research laboratories to enable a broad range of research use or in vitro diagnostic use products for different liquid or tissue biopsy-based applications, where for that particular group or institution, it may make more sense to internalize these capabilities than send out. And so this kind of broad and flexible portfolio of options, we hope will help to bridge the testing gaps that we currently see and identify the most appropriate treatments for patients. So from a drug development perspective, as we think about some of the different ways in which these strategies could be utilized, certainly the most well-established from a patient selection and stratification standpoint now, with multiple FDA-approved companion diagnostics and therapeutic indications for predictive biomarkers based on ctDNA-based assessments. But that's represents one potential opportunity that liquid biopsies can afford. And so the area of focus that we want to discuss today is around evaluation of treatment efficacy. So how can ctDNA quantification be used to evaluate dynamics for molecular response and relapse, both in early stage disease, but also in metastatic settings. And then clonal evolution, or emergence of previously undetected alterations that arise or are clonally selected for under treatment pressure, and how can that potentially be leveraged for improving future combination strategies, or enabling better outcomes for patients who may be initially or develop acquired resistance to a particular therapy. And so as we've thought about expanding liquid biopsy-based applications from targeting individual hotspot alterations within cell-free DNA for mainly for therapeutic selection purposes, and expanding to really all alterations that are detected across these large comprehensive panels, I've started, we and others in this space have started to really see the potential confounding effects of clonal hematopoiesis. So this was a seminal study from 2015, looking in depth at a number of hotspot alterations that really highlighted the prevalence and age distribution of these alterations that can drive clonal hematopoiesis, and really led to some of the findings that we think about today, and that clonal hematopoiesis driven by these leukemia-associated gene mutations can occur without evidence of a blood disorder. And even for this study, which was focused on individual hotspots, clonal hematopoiesis was identified in about 1% of individuals under 60, and almost 20% of individuals that were over 90. And there was a lot of biology that was very interesting, that came out of this study, and several more recently as well, that have highlighted the clonal evolution of some of these different types of alterations. We canonically think about DNMT3A hotspot alterations at codon 882, but you can also see the emergence of spliceosome-related alterations that drive clonal expansion under selective pressure that are more specific to the aging hematopoietic system and are more highly prevalent in older individuals. And all of these taken together are findings that we need to kind of think about and figure out how to most appropriately account for, as we're focusing on somatic but tumor specific alterations within the cell-free DNA compartment. Another study that we were involved with more recently, related to the direct detection of ctDNA with early and late stage cancers, led by Jill Fallon, with the primary objective of evaluating 200 patients with colorectal, breast, lung, or ovarian cancer, and were able to demonstrate detection of ctDNA, anywhere from about 60 to 70% of patients with stage 1 or stage 2 disease. Also found through these analyses, when we were looking specifically at hematopoietic proliferation-related genes, the identification of hotspot alterations, again, associated with DNMT3A, but others that were previously uncharacterized. And interestingly, the levels at which these alterations occurred, were ranged from anywhere from 0.2% to 5.3%, which is often the range in which you expect to see alterations associated with circulating tumor DNA. And so, through the direct analysis of these alterations within cell-free DNA, suggested a higher fraction of asymptomatic individuals could harbor these alterations than had been previously reported, which were focusing mostly on cellular-based assessment of these genes in blood. So, fast forwarding to where we are today, the approach that we've developed for comprehensive genomic profiling from cell-free DNA mainly for therapy selection, but also some of these different applications for monitoring and analyses of clonal evolution for patients with advanced or metastatic disease is PGDx Ilioplasma Complete. It's a 521 gene approach where we evaluate the full coding sequence for these 521 genes for both analyses of small nucleotide variants, insertions and deletions, but also structural changes such as amplifications, translocations, and then common genomic signatures like BTMB and MSI. And so as we've developed this panel, and with the decreasing cost of sequencing, really probably over the past 6 to 12 months, it's now possible to really perform these paired matched white blood cell analyses in a cost-efficient way to accurately differentiate those alterations that may be associated with clonal hematopoiesis, but also rare germline polymorphisms that may not be accurately classified through some of these different bioinformatic approaches that have been developed and optimized to try and enrich for somatic tumor-specific alterations. So a data set that was presented by Tanya Watkins from our group at ACR, highlighted this initial approach where we're able to enhance the detection and classification of cell-free DNA alterations through matched white blood cell analyses. And so the plot on the left highlights the correlation of the cell-free DNA variant allele fraction with the white blood cell-derived variant allele fraction by the different alteration sources. So you can see the alterations that occur at a high variant allele fraction within the matched white blood cells, within the cell-free DNA as well, are typically associated with germline alterations, although they can be highly variable within the cell-free DNA depending on levels of circulating tumor DNA, copy number changes, and loss of heterozygosity at those loci. The alterations that occur at low variant allele fractions within both sample types are associated with clonal hematopoiesis, and then those that are more or less restricted to the cell-free DNA compartment are somatic tumor-specific changes. And then when we compared this to our traditional approaches for characterization of these different classes of alteration on the right, we're able to accurately do that for about 85% of alterations with a cell-free DNA only approach. But this matched white blood cell analysis was able to accurately reclassify about 15% of alterations, which certainly isn't a majority, but given there are multiple alterations observed for any individual patient, can actually affect a larger number of patients than might be expected if you're interested in accurately assigning variant origin for all alterations within a particular sample. So a recent study that we were able to evaluate the improved performance for a matched white blood cell approach is this the Cairo-5 study. So we're fortunately able to partner with the Netherlands Cancer Institute on this, and focused on treatment strategies in colorectal cancer patients with initially unresectable liver-only metastases, where the focus was to randomize patients based on RAS or BRAF mutation status and tumor sidedness to either receive doublet or triplet chemotherapy with bevacizumab or doublet chemotherapy with bevacizumab or panitumumab. And so for the exploratory analyses for this study, we're focused on the panitumumab arm. So for 52 patients within this that were participating in the Cairo-5 clinical trial, we were able to analyze 183 cell-free DNA samples and 49 matched white blood cell samples, along with 28 tumor tissue samples for those patients that had tumor tissue samples available, and analyze those with the PGDX elio tissue complete and PGDX elio plasma resolve assays, with a focus really on identifying tumor-specific plasma cell-free DNA alterations that could be used for evaluating primary and acquired resistance, along with molecular response to treatment. And so initially, we were able to identify a strong correlation between the mutant allele fractions of variants observed in both cell-free DNA and matched white blood cells, accurately classifying those that were tumor-specific, germline, or hematopoietic. And given some of the data that's highlighted, the fragment length for circulating tumor DNA-specific fragments compared to cell-free DNA, we're able to further confirm the accurate classification of these in the bottom figure, looking at the shorter fragment lengths associated with tumor-specific P53 and APC variants compared to those variants that were assigned as germline or hematopoietic status. And so then when we looked at tumor tissue-informed versus white blood cell-informed approaches for assessment of molecular response, there were 46 patients whose ctDNA levels could be determined at baseline. About 61% were classified as molecular responders and 39% as molecular non-responders. And we had matched tissue samples available for 26 of these patients, and were able to compare directly the performance of a tumor tissue-informed approach versus a white blood cell-informed approach in this setting, and found that the molecular response to treatment was less prognostic for overall survival with the tumor tissue-informed approach when ctDNA was not as accurately filtered for germline hematopoietic variants compared to the white blood cell-informed approach. And then for the entire cohort of 46 patients where we were able to perform the matched white blood cell analysis, we're able to compare the cell-free DNA approach to traditional RECIST evaluation of ctDNA imaging, and found a significantly longer overall survival for molecular responders compared to molecular non-responders, compared to patients with or without progressive disease based on RECIST evaluation of CT imaging. And then finally, there was a subset of patients for which we had liquid biopsy samples available post-operatively for the patients that were eligible for surgical resection after downstaging of their solitary liver metastases, and found that the event-free survival was significantly shorter for the 8 patients with detectable ctDNA post-operatively, all of whom eventually went on to experience a recurrence, compared to the 4 patients with undetectable ctDNA after the last surgery, yielding an overall sensitivity of 80% and 100% specificity postoperatively. And then finally, in order to evaluate and identify mechanisms of primary and acquired resistance associated with EGFR therapeutic monoclonal antibody-directed therapy, we were able to evaluate baseline liquid biopsy assessments and identify primary resistance for 17 patients, or about a third of the cohort initially, and then 5 patients, or about 10% of patients, after the initiation of treatment, were mutually exclusive with then the patients who were exhibiting primary resistance, as you might expect. So the combined cell-free DNA and white blood cell analysis prevented false positives due to germline or hematopoietic variants in about 40% of patients. And the patient-matched tumor tissue for the primary analyses that were highlighted didn't provide any additional information for this study. Longitudinal analyses of ctDNA were more predictive of overall survival than standard of care radiologic response. And then primary and acquired resistance to panitumumab was identified in 42% of patients, demonstrating that the accurate calling of ctDNA mutations for treatment response monitoring is feasible in a tumor tissue independent way through this paired analysis of cell-free DNA and matched white blood cells, which has a significant opportunity and simplifies sample logistics to facilitate the application of liquid biopsy-based ctDNA testing for emerging therapeutic resistance, ultimately enabling broader access to how we might be able to think about utilizing and optimizing ctDNA for different types of applications. So now I'll hand it over to Elsa to talk about some of her recent data from longitudinal analyses of ctDNA and for monitoring therapeutic response during immune checkpoint blockade.
Great, thanks, Mark. Hi, everyone. It's a pleasure to be here today. I'm Elsa Anagnostou, and a pleasure to continue the discussion on liquid biopsies. I will refocus our subject to ctDNA response in the context of therapy, and I will specifically focus on ctDNA as an early endpoint for immunotherapy response. Now, Mark already discussed this. Liquid biopsies have been emerging as potentially powerful, non-invasive means of detecting tumor burden, tracking tumor evolution over time. And certainly the clinical utility of liquid biopsies is supported by an ever-growing number of studies that show that circulating cell-free tumor DNA, or ctDNA as we refer to it, can be implemented in detection of minimal residual disease, monitoring of therapeutic response, all this in the context of precision immuno-oncology. And what I wanted to discuss first is, why do we even think about this, right? So why is integration of liquid biopsies a critical step towards precision immuno-oncology? So certainly, as many of us know, optimal patient selection for immunotherapy, that truly represents a challenge. This is intensified by the heterogeneity of clinical responses and certainly the inefficiency of imaging to fully and timely capture a therapeutic response. We're all using in the clinic snapshot biomarkers like PD-L1 expression or tumor mutation burden, but these are truly imperfect. And so what we really need is to develop molecularly informed strategies to, again, enable early and accurate response assessment and then facilitate therapeutic decision-making. Now, can liquid biopsies bring this resolution and precision to immuno-oncology? Of course, this is a rhetoric question. My personal opinion is yes. But before getting to that and discussing the opportunities, I actually wanted to review some of the challenges. And Mark already went through this, so I'm going to go through this very briefly. The key challenge that we have with ctDNA approaches, it's inherent to the scarcity of the analyte that we're actually looking for and detecting. That is ctDNA, right? So those mutated tumor sequences are present in very small quantities relative to normal DNA background. And conventional PCR and sequencing error rate is very similar to those very small levels of mutant DNA. Certainly, over the past decade, we've seen the development of several next-generation sequencing technologies for detection of mutations in ctDNA. These employ deep and redundant sequencing together with implementation of nucleotide barcodes, sequencing error suppression algorithms, multi-mutation tracking, the panel approach. Together, all these steps allow for the sensitive and specific detection of mutant sequences from cell-free DNA. And so what I'm showing on the left-hand side here is a hybrid capture-based approach. This is very much what Mark, or most of what Mark was showing previously. And what I'm showing on the right is a tumor-informed multiplex PCR approach. The error rate with these approaches, and I'm showing that on the bottom left here, is several orders of magnitude lower compared to conventional sequencing. So certainly these are breakthroughs that have allowed, technological breakthroughs that have allowed for the sensitive and specific detection of mutations in ctDNA. Now, let me very briefly review biological noise. Mark very nicely reviewed the confounding factor of clonal hematopoiesis. It is such an important issue, and I wanted to emphasize again here. And so, just for everyone to be reminded of the fact that the majority of cell-free DNA in plasma is released from hematopoietic cells, not from tumor cells. So any blood-based NGS approach has the potential to detect clonal hematopoiesis mutations, and these may be classified or misclassified rather as tumor-specific. And Mark mentioned this, but up to 50% of cell-free DNA mutations may be of clonal hematopoiesis or germline in origin. And to make this more complex, these mutations from white blood cells can fall in genes not necessarily canonically associated with clonal hematopoiesis, right? So we don't see these mutations always fall in DNMT3A or P53 or other canonically mutated clonal hematopoiesis genes. And again, this highlights the importance of developing ways to carefully characterize the cellular origin of mutations that are detected in liquid biopsies. Now, having very briefly discussed the capabilities and limitations of liquid biopsies, let's focus on the clinical implementation of these approaches in measuring tumor burden. And as I'm showing you here, ctDNA can be tracked longitudinally for minimal residual disease, or MRD detection. This is the early stage paradigm that I'm showing on the left, as well as response monitoring. This is the metastatic paradigm that I'm showing on the right. And I will discuss the metastatic setting first. So in this schematic, we're summarizing the approaches for the detection and analysis of ctDNA now in the context of immunotherapy, again, focusing on the metastatic setting first. For the most part, what we're using in this setting is tumor agnostic approaches, and integration of ctDNA analysis is truly important for immuno-oncology, for potentially patient selection for immunotherapy, subsequent response monitoring, especially in particular settings, and I'm going to call them out and get back to them. But one setting that ctDNA approaches can be particularly informative is for patients with radiographically stable disease, or with patients that response radiographic response is a little bit more challenging. For example, patients with mesothelioma that is tough to capture with radiographic imaging. Overall, accurate therapeutic response monitoring by liquid biopsies, as we will discuss a little bit further later, can inform decisions on the optimal duration of immunotherapy, as well as inform appropriate strategies for treatment escalation or de-escalation based on ctDNA molecular responses. Now, how about using liquid biopsies to complement tissue biomarkers in predicting therapeutic response in the context of immunotherapy, especially in cases where tissue biomarkers fail to do so? And an example that I'm giving here is the challenges that we're facing with tissue-derived tumor mutation burden. That, of course, is the prototypic biomarker of response to cancer immunotherapy. However, we all know that TMB is an imperfect biomarker of response that is affected by technical and biological limitations. And what I'm showing you here is a technical limitation that has to do with underestimation of the true tumor mutation burden in the context of low tumor purity. So what we're showing here is in silico dilution, an in silico dilution experiment using a clonally complex TMB high tumor. We're showing that in the top row, and a clonally homogeneous, you can see just two clonal clusters, TMB low tumor. We're showing that in the bottom row. And what you'll appreciate if you look at the far right, the power of detection of mutations significantly decreases with decreasing tumor purity. And what happens if you focus on kind of the middle of the in silico dilution experiment that we're showing here, at the lower end of tumor purity, say 40% or below, TMB is of a clonally heterogeneous TMB high and a clonally homogeneous TMB low tumor. These become indiscernible. So that's a challenge, right? Now, could blood-derived TMB remedy this issue? Is certainly conceptually, ctDNA is the pool of ctDNA is representative of mutations originating from distinct primary and metastatic tumor sites that may better capture tumor clonal heterogeneity. And certainly we were very excited when retrospective studies started coming out showing that there may be a predictive potential for blood TMB. We saw that in retrospective assessments in the Poplar, the Oak, the Mystic trials of atezolizumab in non-small cell lung cancer. But when we moved to prospective analysis using pre-specified blood TMB thresholds, these did not confirm the predictive value of blood TMB. So two representative studies are projected here. The BEFIRST study is shown on the left. This is a study of first-line atezolizumab in patients with metastatic non-small cell lung cancer, where a blood TMB of 16 mutations per megabase was associated with a high overall response rate. But as you can appreciate, with in the survival curves, these findings did not translate at a significant difference in progression-free and overall survival. And the same holds true for the BFAST study that we're showing on the right-hand side. So there are certainly several technical and biological limitations that have to be considered here, and perhaps have to do with lack of algorithm standardization, threshold harmonization for blood TMB, and, as Mark brought up, accurate exclusion of alterations that may be associated with clonal hematopoiesis. Now, moving on to dynamics, ctDNA dynamics, this, in my opinion, represents the most promising biomarker of response in the context of immunotherapy for patients with metastatic disease. So we and others have applied ultrasensitive next-generation sequencing methods to analyze ctDNA in serial blood samples. Here, a lot of studies focus on patients with lung cancer during immune checkpoint blockade therapy. And collectively, these analyses have revealed very distinct patterns of ctDNA-based molecular responses that are actually reflective of patients' clinical outcome. And you can appreciate those on the right-hand side of this slide. So patients with a ctDNA molecular response have a dramatic reduction in ctDNA to undetectable levels, which is reflective of long-term clinical benefit. And in contrast, as you're seeing on the bottom right there, patients with primary resistance to immunotherapy, ctDNA levels truly have limited fluctuations or actually display a rise during therapy. Now, accurate determination, I cannot stress this enough, accurate determination of variant cell or origin is very important in assessing ctDNA molecular response in the context of immunotherapy and any therapy, really. Again, a good fraction of alterations detected in plasma NGS are clonal hematopoiesis related. And one way to overcome this issue, as we did in this study, would be to perform matched leukocyte DNA sequencing and filter out in that way clonal hematopoiesis variants. And that approach would allow for accurate classification of plasma variants in clonal hematopoiesis and tumor-derived categories. That's the first split. And then focus on the tumor-derived variants that can in turn allow for assessment of ctDNA dynamics and molecular response. Now, after we do that, after we classify variants by cell or origin, we can then start to look at the value of ctDNA in interpreting heterogeneous clinical outcomes. And as I brought up before, ctDNA molecular responses may be particularly informative in patients with radiographic stable disease, which represent a heterogeneous group. And as you can appreciate in the top graph and then the very bottom on the left, ctDNA molecular response was actually not concordant in this study with first radiographic response, while there was a trend noted towards a correlation between ctDNA response and best overall radiographic response. And what is interesting and of course expected is that ctDNA molecular responses were concordant with durable clinical benefit in the context of immunotherapy. And that now, if we look on the right-hand side, this is reflected in ctDNA molecular response, more accurately distinguishing progression, patients with longer progression-free and overall survival within this very heterogeneous group of patients binned in a stable disease category radiographically. Now, the association between ctDNA response and improved survival, this has been supported by meta-analysis or pooled analysis. This was, I'm showing here a representative example. This is a CT monitor project from Friends of Cancer Research. And in this initiative, ctDNA kinetics from smaller IO-treated non-small cell lung cancer cohorts, these were harmonized, and the association between ctDNA dynamics and long-term outcomes was assessed in the larger pooled data set. Despite the cohort and methodology heterogeneity, again, here we saw a significant association between ctDNA reduction and better outcomes with respect to overall progression-free as well as overall response rate. Now, this is all great, and the association of ctDNA response has been reported in several proof-of-concept studies, as I showed. However, there are still outstanding questions that have to do with the definition first, the definition of ctDNA response. So what is ctDNA response? And I would argue that the definition of molecular response is actually ctDNA assay specific. So here I'm showing you a variation in mutant allele frequency replicates of tumor-specific mutations. This is a mixture of DNA from tumor cells spiked into wild-type DNA. And what you'll appreciate is if you focus on the middle panel there is that the coefficient of variation increases with decrease in mutant allele frequency, and certainly the uncertainty around that MAF estimate increases as we go down to lower mutant allele frequencies. And all that these analyses say is, now if you look on the right hand side and take a MAF of 1%, we have a line there. The 95% uncertainty in this MAF estimate ranges from minus 50 to 50%, which is 0.5% MAF to 1.5% MAF, right? So in this case, or in such cases, a greater relative reduction is required to determine ctDNA response. And that's where complete ctDNA clearance comes in, which is a definition that we have adopted in terms of determining ctDNA molecular response. Another open question, when does ctDNA response occur? And in the IO space, we know that this is somewhere between 4 to 9 weeks, with reliable ctDNA responses seen as early as after 1 cycle of immune checkpoint blockade. And certainly, major question, what is the concordance between ctDNA molecular response and radiographic response? We need to know this in order to design the ctDNA interventional clinical trials. And as I'm showing you here, if you focus on the top left, where this is a reanalysis of published or soon to be published studies that is close to 80%. It can be lower, and this can be driven by the differential ctDNA responses in patients with radiographically stable disease, as we discussed. But if we look on the right-hand side and looking at long-term clinical outcomes, it is apparent that ctDNA response better captures longer overall survival independent of the radiographic response. So how do we do this? How do we address these open questions? How do we gain more granularity and move from proof of concept, right, or pilot studies or low resolution, if you will, to high resolution? So the answer here, I think, is to establish the value of ctDNA as an early endpoint of immunotherapy response in clinical trials. And to answer these questions and test ctDNA as an early endpoint of response to immunotherapy in a molecular response adaptive clinical trial setting, we designed BR36, the BR36 clinical trial, which is conducted in 2 stages. I'm showing you here the lead-in initial stage that was particularly designed to answer the questions that I posed before, which were what is the definition of ctDNA molecular response, when does it happen, and what is the concordance with radiographic RECIST response. Again, the setting here was patients with metastatic non-small cell lung cancer that were PD-L1 positive, oncogene negative, that were treated with pembrolizumab as standard of care, for which we performed serial liquid biopsies with each cycle of pembrolizumab monotherapy. And overall, what we learned from the first stage of BR36 is that ctDNA response, and I'm showing that on the left-hand side, this is very distinct in terms of the pattern that we see in blood, in plasma, from molecular disease progression that we're showing on the right. And ctDNA response, roughly at 6 weeks during pembrolizumab monotherapy, can identify patients with molecular disease progression that can be randomized to intensified therapy, while you can imagine that patients with molecular response can be spared. And this is important, and I cannot stress this enough. It's important to understand concordance or discordance with radiographic responses, and dissecting this concordance or discordance between molecular and radiographic responses and how these actually capture long-term outcomes. This is crucial in moving on to ctDNA response adaptive clinical trial design. Now, how do we implement ctDNA response in clinical trial design, right? So we talked about this, it's important. How do we do it? It can happen several ways, right? We can implement ctDNA in treatment de-escalation in the setting of sustained ctDNA clearance. I'm showing that at the top left here. This is something that is less studied, so we don't really have the answers in terms of how specifically to do that and when to determine potential de-escalation of therapy based on ctDNA. But ctDNA can also inform early switch in the setting of recrudescence or emergence. In this, may have a role in refining drug development strategies and establishing ctDNA as an early endpoint to support drug approvals. The longest hanging fruit certainly is what I'm showing in the bottom right there, which we've already talked about, which is the following: that liquid biopsies can inform quick fail approaches, right, where patients with ctDNA progression are rapidly and accurately identified and channeled towards different therapies. So I wanted to spend a few minutes very briefly to discuss the role of ctDNA tracking for minimal residual disease monitoring in the early stage setting. This is now relevant in the IO space, as of course immune checkpoint blockade has been implemented in neoadjuvant therapy for resectable non-small cell lung cancer. And wanted to highlight the emerging data in this context, where liquid biopsies and ctDNA dynamics or status predicts pathological response to neoadjuvant immunotherapy. This is Checkmate 816, the neoadjuvant lung cancer immunotherapy trial, where we learned that patients with early stage non-small cell lung cancer receiving adjuvant nivolumab and chemotherapy that also showed ctDNA clearance preoperatively, these patients had a longer regression-free or event-free survival and were more likely to attain a pathological complete response, as we're showing them in the green bar on the right-hand side. Now, this is truly an evolving field, but I wanted to make a point with respect to the accuracy of ctDNA detection for pathological complete response with neoadjuvant immunotherapy. The way we're computing the clinical sensitivity here is the fraction of patients that attain a path CR that also have undetectable ctDNA. And what you'll see here is that the sensitivity is actually good, but specificity is variable, and that may be, and this is driven by the fact that there's a sizable fraction of patients with undetectable ctDNA that actually do not attain a pathologic complete response. And that may not necessarily point towards a worse prognosis, rather shows a challenge with path CR as an endpoint. So more to come. This is an exciting and evolving field. And so collectively, everything that I showed you today and I discussed, and both Mark and I discussed, these efforts are tailored towards delivering the best available clinical care with precision. I cannot stress this enough. We need to, in order to be able to test the true potential of liquid biopsies, we need to implement these approaches more and more into clinical trial design. So just for the last few minutes, I wanted to bridge everything that it is we talked about, which is more so in kind of the research or now budding clinical trial setting. I wanted to bridge to immediate clinical applications and stress and highlight again what Mark already mentioned, that ctDNA or liquid biopsies offer a complementary tool for capturing evolution of resistance mechanisms to cancer therapy. And this was very nicely shown in the study that we're projecting here, where you can appreciate the increasing discordance between archival tumor and liquid biopsies with the time lapse between tissue and ctDNA testing, and an increase in the proportion of patients with higher numbers of tumor-derived mutations in liquid biopsies compared to the original tumor biopsy. So how do we use this data? I wanted to leave you with a very pragmatic trial that we are doing within the Johns Hopkins Molecular Tumor Board, and Labcorp is a sponsor here, where we're using liquid biopsies together with a rigorous mutation actionability characterization approach to match genotypes from plasma NGS with molecularly driven clinical trials, with the primary objective to validate the feasibility of the approach in informing clinical decision-making. So let's go through our take-home messages on my end. So certainly liquid biopsy approaches are being tested in a variety of clinical diagnostic applications in precision oncology and precision immuno-oncology in particular. ctDNA kinetics and molecular response in the metastatic setting represents a very promising early endpoint of response. It's now starting to be tested in clinical trials. Just a word of caution, it is important, very important, to select the liquid biopsy approach based on the diagnostic application and intended use. It's important to make sure that biologically relevant alterations are considered. The technical performance of the approach is sufficient to minimize error, and certainly consider the detection and filtering of clonal hematopoiesis mutations that we discussed today. I'm going to end again with call for clinical trials. We certainly need additional studies in the context of clinical trials to further support the implementation of liquid biopsies in clinical cancer care. And stop here and highlight an educational resource that we put together with Mark Stewart from Friends of Cancer Research, and Mark Sausen has worked with us in this initiative as well. This goes over several of the aspects that we discussed here today, also includes regulatory perspective in terms of the evidentiary roadmap, if you will, to implementation of liquid biopsies. So you can access this resource as shown on the screen. So thank you all, and looking forward to questions.
All right. Great. Well, thank you, Elsa, and thank you, Mark, as well, for your informative presentations. We will now start the live Q&A portion of the webinar. So if you have a question that you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, type in your answer, click Submit, and we'll answer as many of your questions as we have time for. All right, let's get started. Our first question here asks, how can we accelerate more rapid adoption of liquid biopsy-based testing as a routine diagnostic approach?
Yeah, sure. So I'm happy to maybe start that one. I think as Elsa highlighted, I think the most robust way to implement liquid biopsy-based approaches and have that lead to a change in standard of care is through clinical trials. And I think for liquid biopsy, based assessments, whether that's tumor profiling or for these analyses of molecular response, early endpoints. I think it could be a combination of well-designed observational studies. If there's still a need to kind of further optimize a particular signature, better understand clinical sensitivity or specificity, and association with other clinical pathologic factors, where in many cases ctDNA may not necessarily be a completely independent biomarker, but is going to be viewed in the context of other factors that may be used for how to manage a patient's care. So observational studies can be great in that setting. I think the BR36 study Elsa highlighted is a very good example of that, where there were a couple of key questions that needed to be addressed before you'd be necessarily comfortable moving into an interventional study that is ultimately needed to demonstrate the improved outcomes associated with some of these different approaches. So I will also echo the call for clinical trials, both observational and interventional, depending on the specific platform, biomarker, and application. But Elsa, do you want to add on to that as well?
Yeah, absolutely. I think, again, cannot stress enough that we truly need to be implementing molecularly informed approaches into clinical trial design, right? So we know in the context of immunotherapy, imaging is truly inefficient, especially in the context of stable disease, right? So doesn't capture the nature, doesn't capture the magnitude of therapeutic effect. Certainly the timing is off, right? So we need to be doing this. I also want to bring up here kind of the different applications of liquid biopsies, right? So certainly if one talks about ctDNA response or minimal residual disease tracking, right? So we need to prove their clinical utility in the context of a trial, right? But just to remind everyone that plasma or liquid biopsies or plasma NGS for genotyping, this is a very viable approach in clinical practice right now that is integrated in kind of our routine clinical care, for the most part as reflex testing if tumor NGS is not yielding the results that one is anticipating. But there is increasing efforts, for plasma first, right? So for plasma first approaches, and Mark discussed those or some of those in his presentation as well. just a word of caution here in terms of sensitivity of liquid biopsies, right? So the technical challenges, I think we're doing better as a field with new and emerging technologies, but a clinical challenge that we're facing is when the liquid biopsy comes back and it's negative, right? What does that really mean? Does it mean that, the tumor doesn't really harbor that EGFR actionable alteration? So these are, I think, implementing liquid biopsies into kind of our clinical diagnostics approaches in a thoughtful way, and probably tiered and combined approach with tumor biopsies for genotyping now, right? Is an important approach, and we actually have started seeing these initiatives being implemented.
Great, thank you both for that answer. And I know that we are running a little bit short on time, but we do have time for a few more questions here. So this next one asks, what do you think about droplet digital PCR for low abundance mutations detection in ctDNA as compared to NGS approaches? Is it better or more sensitive?
Yeah, perhaps I can start with this, and then Mark, you can weigh in, on the technical side as well. But conceptually, certainly, genotyping the tumor and then tracking specific tumor-derived alterations by approaches, say, like droplet digital PCR, that can allow for very sensitive detection of these particular mutations. I think. And that certainly clinically is very meaningful, especially if one is thinking about minimal residual disease detection, right, in the context of early stage disease. But a point that I want to make here is the fact that these approaches, we call them bespoke, right? So tumor informed, where you have to define the alteration of choice in the tumor and then track it in plasma, they may not be that visible in clinical practice. If we want to scale this up, we want to implement ctDNA MRD, right? Can we truly make an assay for every single patient, right, that we see? So there is a lot of, this is where kind of the tumor agnostic approaches come in. Certainly multi-gene panel approaches increase the sensitivity because you're not just tracking one mutation, you can track multiple. So that's I think it's a sensitive approach. It may not be a feasible approach for broad clinical integration.
Yeah, and maybe just to add on that, I think I think those are great points. I think there's it may be variable depending on the application. So I think a lot of the early ctDNA work was really done through digital PCR based approaches. Led to a lot of seminal findings. Given the sensitivity and specificity of those types of technologies. As we think about implementing these for a clinical use. Then I think also made some great points around challenges associated with utilization of those prospectively, potential to incorporate the matched tumor. But I think is also somewhat depends on the application as well. So if there's an interest in looking at molecular response and you demonstrate molecular progression, then there may be a question around well what specific alterations are present within that patient's tumor, and then a broader panel-based approach that's guided through a matched white blood cell analysis may potentially have additional value to be able to identify both the molecular progression as well as potentially additional alterations that may be considered for how that patient's care could be managed. But great question, and I think maybe is complementary to how you might consider using panel-based approaches.
Great, thank you. All right. Well, that unfortunately will conclude our session for today due to our time limit here. So I do want to thank you again, Mark and Elsa, for your time today and your important research. We would also like to thank Labroots and our sponsor, LabCorp, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.
Goodbye.